So, can you briefly describe like the type of research that goes on in your lab specifically right now?
Sure. So in my lab, we work at the interface of two different kind of research areas, which is we have nanomedicine and we have tissue engineering, and we have projects in both of these areas basically. So in the nanomedicine area, we are focused on developing inhalable nanoparticle formulations that can be used to treat diseases in the lungs. So this could be lung cancer or lung fibrosis or COPD or things like that. And we develop multifunctional nanoparticles. So basically one formulation that can be used maybe to deliver two different kinds of therapies at different rates. So basically providing combination therapy, or you can incorporate an imaging agent so that you can image where these nanoparticles are going after they are administered. Or you can have a targeting component. So you can have these particles go and bind to specific cells so that the therapy is given only to the cell that we are trying to treat. It could be fibroblasts or epithelial cells or endothelial cells. So things like that. And then we have the tissue engineering component where we develop disease models in the lab. Because I'm sure everyone has seen people working with cells growing in petri dishes in the lab and using these cells to test different kinds of therapies. But you know that cells in the body are not present as just a flat two-dimensional layer. You see them as complex cell aggregates. So in the lab, we are trying to mimic those kinds of conditions, those cell arrangements, so that we can mimic how the disease is inside the body, so that when we then use these cell aggregates for testing different nanoparticles or different therapies, then the responses of these cells will be more accurate as well. Because in the last few years, when people have increasingly started to realize that when you grow cells as a flat layer in well plates or petri dishes or flasks, those cells, they don't respond the way cells in the body respond to therapies. So we are in dire need of better models in the lab. So we are focusing on developing these tissue engineered disease models for testing all these nanoparticles and other treatments as well.
What are some specific techniques or practices that you utilize in your lab, like the DLS and all that type of stuff?
Yes. So for the nanoparticle work, we use the DLS, Dynamic Light Scattering Instrument, which can be used for measuring particle sizes, how stable these particles are in solutions, how uniform their sizes are in the solutions, things like that. Then we have we use different techniques for lowering the sizes of these particles. We need to make them in the nanometer range, obviously, and it needs to be at least less than 200 nanometers. 100 nanometers is perfect, but at least less than 200 nanometers. So we use things like the sonicator, which can be used to break down our nanoparticle formulation so that they're really small. So we also use the confocal microscopy and EVOS images, which are basically used for seeing where these particles, we prepare fluorescent particles and we see where they end up inside the cell, whether they go to the nucleus, whether they stay in the cytoplasm, whether they're bound to the surface of the cells. That can be visualized with confocal microscopy. And then in the tissue engineering area, we have recently started working with 3D bioprinters. So we have two 3D printers in the lab that is being used for training students in the classroom on different methods of developing tissue engineered organs. And so as an example, so I have this prop that I use quite often, which is of a 3D printed brain. And this is actually made with plastic. But we can, with the 3D bioprinters in the lab, we can develop this model using actual brain cells. And we can then culture like a three-dimensional brain tissue in the lab, as I said before, for testing different kinds of therapies and for modeling different kinds of diseases. And the other thing we have, and maybe you've seen this already, so we have this very futuristic looking couple of devices in the cell culture space. So we have this orb, which, I think somebody told me it looks a bit like an alien spaceship sort of thing. And what it does is it's used for culturing cells in different conditions within something called as organ chips. So this is an organ chip, and you can see that there are two channels. There's a pink channel and a blue channel in this, right? And what we can do is we can culture different cells in the different channels. So for example, you can grow endothelial cells mimicking the blood vessels in one channel. We can culture epithelial cells in another channel. And using this orb, we can mimic things like, for example, mechanical stresses. If you want to make a lung on a chip, you can introduce mechanical stresses, so you can expand and contract this chip. So the cells here are going to experience the same stresses that cells in the lung would experience during breathing. You can introduce fluid flow in these channels so that you can mimic how blood flows through the blood vessels, and what that flow and what that shear stress does to the cells, the endothelial cells in the blood vessels. So all of that can be mimicked with these organ chips. So and then we have technologies that we're developing in the lab. So we have microwell arrays, which are gels with little micrometer sized wells in which we can culture cells, and these cells will be in a 3-dimensional format. And then we have scaffolds. We make porous polymer scaffolds on which cells can be cultured. So our aim with all of these technologies is basically finding the best method to mimic condition, like disease conditions in the lab, whether it's introducing fluid flow, whether it's introducing stresses, whether it's introducing a substrate for cells to grow on. And if you're doing lung tissue engineering, then obviously the epithelial cells need to be exposed to air. So how do you do an air-liquid interface for these cells? So all of this is work that we're doing in the lab right now, and basically trying to find the best way to mimic all these diseases in the lab.